I would like to introduce our first presenter today, Leyla Kaufman. And just a heads up, this is a little different. We have two presentations back to back today. Leyla will be presenting first, and then we're going to have Mark Wright presenting right after her. And we'll be having questions right after Leyla's talks, like probably about 5 to 10 minutes of questions, and then we'll jump into Mark Wright's presentation. So for those of you that don't know Leyla, she is the program coordinator for the Māmalu Poepoe Biosecurity Program. She has a background in entomology. She's studied biocontrol in Switzerland and has dedicated her research to agricultural and forest pests here in Hawaii. She is born and raised in Peru, and I'm really happy she can be here today to present about the Māmalu Poepoe Program. So Leyla, I am going to hand it over to you to share your screen, and I will go on mute and stop video. And just for the audience, feel free along the way to type in any questions. You can type them into the chat box, or you can type them into the Q&A. And Elizabeth and I will be happy to share those questions at the end of Leyla's talk. So again, I'm Chelsea with Hawaiʻi Invasive Species Council, and we have Elizabeth here who runs and operates the 643 Pest Hotline. Okay, Leyla.
Thank you so much for the introduction. Welcome everybody. I'm Leyla Kaufman. I am the program coordinator for Māmalu Poepoe, and I hope you can see my screen right now. Is that?
Yeah, looks good.
Great. So Māmalu Poepoe is an airport surveillance program. It's a biosecurity airport surveillance program. And before I dive into what Māmalu Poepoe is, since we are in this biosecurity theme talks, this one and the next one, I wanted to provide a definition of biosecurity. So biosecurity is the set of measures taken to prevent and to manage the risk from invasive species to the economy, environment, and health and lifestyle of the people. And this definition is actually taken from the Hawaii Interagency Biosecurity Plan. This plan, I'm pretty sure you are very aware of this plan, and this was adopted in Hawaii in 2017 and is a coordinated path forward for different state agencies to increase support for agriculture, the environment, and the health and lifestyle of local people. And this interagency biosecurity plan lays out a number of actions. There are 147 actions that basically guide the process to have a more biosecure Hawaii. So in this sense, the Hawaii Invasive Species Council, as well as the coordinating group of alien pest species, launched a joint 2025 strategy in support of the Hawaii Interagency Biosecurity Plan. And this was built on the foundation of this Interagency Biosecurity Plan. This looks at what the HISC and CGAPS networks can do, whether individually or together, to be able to support this biosecurity plan, and identifies Māmalu Poepoe as an important program for early detection and rapid response. Actually, in the Interagency Biosecurity Plan, Māmalu Poepoe is not listed there because it was not. It was still in the works when the plan was written. But the HISC and CGAPS joint strategy do have Māmalu Poepoe as an important program to support. So before I go in and talk about Māmalu Poepoe, I just wanted to show this slide, and this is also taken from the Interagency Biosecurity Plan, and this is the Biosecurity Continuum, and this shows the stages of biosecurity, pre-border biosecurity, border biosecurity, and post-border biosecurity. So for the pre-border, we have the policies and processes to prevent invasive species from making their way to Hawaii. So these are offshore things that we can do to prevent bad things to get here in the first place. border biosecurity, policies and processes that support inspecting incoming things, for example, quarantining things, inspections at the border, and establishing trapping and surveillance program, like, for example, Māmalu Poepoe that works at ports of entry at airports. And finally, that post-border biosecurity, that are the policies and processes that support detecting and responding to incursions and controlling established species. So here is when things already are past the border. They can be maybe in the early detection, rapid response phase, and things that we can do to manage those invasive species. And there have been talks about things that the community can do to help in this post-border biosecurity, like, for example, the 643 pest online reporting, that there was a talk on that last week by one of our HISC staff. So you can go back to those talks and check those out. They are all recorded. And there will be a talk also after mine that talks about biological control. So this is another tool that we have to manage invasive species that are established already and that are causing significant impacts in the environment. So this is another way to see the timeline of invasive species. And this is the invasive species, the species invasion curve. I'm pretty sure you have seen this already many times during these HISC talks. So I won't really go in detail on that, but basically it shows the timeline of invasion, like time in the x-axis, the area occupied by an invasive species in the y-axis. And you can see all the different stages of the invasion, from prevention, like things that are not here yet, they are absent. So we want to be in this kind of like left side of the curve as much as possible, because these are certainly the most cost-effective way of dealing with invasive species. So here is the prevention. Here is kind of like the eradication part of the entry of species is right here. And this is where Māmalu Poepoe operates, right at ports of entry at airports at the moment. So, and you have this kind of window of early detection and rapid response that you don't want to miss. And because the more time it passes, the more the populations start growing and start establishing in their new environment, and then it's more expensive to deal with them. So now that I'm done with those kind of like terms and giving you some background, I will be talking to you about the program I coordinate that is called Māmalu Poepoe. And if you are not aware, or if you don't know Hawaiian language, it means sphere or circle of protection. And this is an interagency airports monitoring project that was launched in 2016. And some of you might wonder why we have a project like that. And it was basically to fill gaps in invasive species surveillance that we had at ports of entry. And this gap was because of the 2009 economic downturn that we had at the time, that caused a lot of furloughs and layoffs. So agencies like the Department of Health and the Department of Agriculture lost that capacity, and therefore were not monitoring airports for key invasive species. So that's when talks started and discussions about launching a project that can fill that gap. So this project brings together 5 different agencies: the Hawaii Department of Transportation, that is also the funding agency for this project, the LNR, the Hawaii Department of Agriculture, the University of Hawaii, and the Department of Health. So this is an airports surveillance program that currently targets 6 main airports statewide: Lihue Airport on Kauai, Daniel K. Inouye International Airport on Oahu, Molokai Airport on Molokai, Kahului Airport on Maui, and Kona and Hilo International Airport on the Big Island. So we have different targets under this program. So we have mosquitoes, especially we are targeting mosquitoes in the genera Aedes, Culex, and Anopheles, Africanized bees, the coconut rhinoceros beetle. There will be a talk on that at noon, so tune in for that one. As well as ants, and especially the red imported fire ant, Solenopsis invicta, and Wasmannia auropunctata, that is the little fire ant. There was another talk on their fire ant last week by a HISC staff. So if you're interested in that target, you can tune in and go back to the recording and check that out. So I will be going through each of these targets in a little bit more detail and tell you why we have these targets under this program. Mosquitoes first. Why? Because mosquitoes, the introduction of any new species of mosquitoes in any new area is considered a public health risk because they can transmit many human diseases. And here I list just a very few, like dengue, chikungunya, West Nile virus, Zika virus, malaria. So we had in Hawaii some dengue outbreaks in the past, like the last one was 2015, 2016, and again, before that one, 2011, I believe, on Oahu. So, so we know, like, during those times, it's kind of difficult to our lifestyle during those times, and we don't want new things coming in, like Anopheles mosquitoes that can transmit malaria. Because if you feel like now, for example, going around with a mask is a change of lifestyle, like I think sleeping under a net can be also like a drag if we get to that point. So if you didn't know, there are over 3,000 species of mosquitoes worldwide. Of those, around 200 species are already known to occur in the continental US. Some of them are actually native to the continental US, but many of them are non-native species. The good news is that we only have 6 species of mosquitoes already established here in the islands, and these are the species that we have here so far. Aedes albopictus is everywhere. Aedes aegypti, that is a very effective vector of dengue, for example, is restricted so far to the Big Island, but has been intercepted many times at Honolulu International Airport, the Daniel K. Inouye International Airport. The last time it was intercepted was, I think, in August 2014. So we haven't had an interception for a while, and we want to keep it that way. It was also intercepted on Maui, on Kahului Airport, back in November 2017 as part of these activities from Māmalu Poepoe. Luckily, it's not established there. It was just an isolated event. And we don't have Anopheles mosquitoes in Hawaii, so which is a good thing because they are the ones that transmit malaria. But one specimen was intercepted back in 2003. I believe it was at a port of entry. I don't know if an airport or a harbor, but we haven't had any interceptions since then. So what do we do at airports? So we set up different kinds of traps that can hopefully attract many different species as they come. So we are just trying to, of course, to monitor for any already existing mosquito here in the islands, but also our main goal is to be able to intercept any new species that get to our borders. So we use OV traps, we use gravity traps, GAT traps, CDC light traps, BG sentinel traps. And these are just pictures of vector control workers as well as staff from the Maui Invasive Species Council is setting traps at ports of entry. We also target Africanized bees. Why do we target these species? Because these are known as killer bees. So these are a very aggressive race of honeybees that we certainly don't want here in Hawaii. And they are adapted to tropical warm climates. So they will thrive if they get here. They are already established in the US mainland. And many occur in the southern states where we get our goods shipped from. So it's definitely a risk. And we, the state intercepted a container that actually had Africanized honeybees. It was, I think, a container that had pharmaceuticals that when an inspector opened it, it realized that there was a swarm of bees inside. So it quickly closed it, fumigated, called the Department of Agriculture, and they took samples, and the samples show that it actually came out positive for Africanized honeybees. So we were certainly very close to having those Africanized bees here. So we don't want them here because it can devastate our beekeeping industry. So the beekeeping industry is big here in Hawaii. And this is all not only the production of honey, but the pollination for crops, as well as our production of queen bees. That Hawaii is actually is a good producer of queen bees, and we provide these queen bees to the mainland, and they fill an important role in mainland agriculture. And of course, the incursion of Africanized honeybees can also affect our tourism industry and our local lifestyle. So this is what we do at airports to monitor for Africanized honeybees. We have these swarm traps. They are corrugated paper traps that we have a lure inside, so it will attract any swarm that lands nearby. And these are pictures of the different HISC staff, like this is specifically HISC staff, the Hawaiʻi Invasive Species committee staff, like setting up a trap and retrieving a trap that has bees. And I know so many of you really love bees, but we have no tolerance for bees that are ports of entry because they are potentially Africanized bees. So we do kill these bees. We don't relocate any bees that are intercepted or nearby ports of entry. So we freeze them, we take samples, we make sure they come out negative for Africanized honeybees, and we go through the contents also to make sure there are no any new bee pests coming to the state or to a specific island. Because, for example, varroa mite, a very important pest of bees, is currently not found on Kauai or Maui Nui. So hopefully they don't get their way there anytime soon. We also target invasive ants, and as with mosquitoes, there is no native ant species in Hawaii. There are currently over 60 species of ants already established. This program mainly targets the little fire ant here and the red important fire ant because they can really have negative impacts to human health, native biodiversity, and the local economy. Actually, a study done little fire ants show that the state spends roughly like $200 million a year on Hawaii Island alone to manage little fire ants. Another study also showed that if RIFA, the red imported fire ant, is to become established in Hawaii, we'll probably be spending the same for managing these invasive species. So this is what we do at airports. We survey with baited vials to detect invasive ants. We use different lures, like a protein lure, like Spam and peanut butter, and the other one has jelly, so a carbohydrate. And this picture just shows stuff from the Hawaiʻi Invasive, the Hawaiian lab, doing a survey at HNL. The coconut rhinoceros beetle, there will be a talk about that later on. So it was detected in Hawaii in 2013. It's currently restricted to Oahu. So, and the CRB response is doing a really great job trying to contain and eradicate these invasive species that attacks palms and coconut palms in particular. And coconut palms hold a great cultural and economic significance and is widely recognized as a symbol for the state. There is not really an extensive economic study done on CRB so far, but a study done by USDA AFS back in 2018 actually reported that the replacement cost of coconut palms in public beach parks will cost approximately $3 million annually. So that is a big expense just if you are just focusing on beach parks. And these are the kind of traps that we have at the airport. I'm pretty sure many of you are also familiar with these traps because they are everywhere. So if you see anything, any activity in those traps, or even if you see damaged traps, it will be good that you report those. There is a number in those traps, and I believe it's the 643 pest number in there. And this is just a picture of the HISC staff, the Maui Invasive Species Committee staff, setting up a CRB trap at Kahului Airport. I'm pretty sure you have seen already these pictures, but I'm just showing it again. This is picture of Waikiki and the wonderful coconut palms in there. And now that we can see them that way, but if CRB really impacted these coconut palms, then they can end up looking like this. I know you can say, well, this is very dramatic, but this is actually not very far from the truth, because coconut palms are looking like that in Guam, where the CRB really kind of like got out of hand. So now that I went through the main targets of Māmalu Poepoe, so this is Māmalu Poepoe is airport surveillance program, but also serves as a platform for collaboration. This program has a working group that meets twice a year, provides guidance to the group. We talk about findings during the last 6 months. We talk about future things that we will be doing. So, and it's a great opportunity to hear from project partners, and they provide feedback on that as well. This program capitalizes on existing capacity from project partners. So we partner with the different invasive species committees, as I said. So like HISC or HISC here on Oahu, MOMISC, MISC, and BISC, and the Hawaiian lab. So because of this partnership, we are able to cost share a lot of the expense. This an economic study actually showed that the actual cost of the program is nearly $600,000 a year, but the actual the cost that we fund is only a fraction of that. Is in the 140s, just because the other part of the expense is cost share with these partnerships. The program provides funding opportunities, opportunities to train partners. Like we provided training for the Department of Agriculture staff to do training on Africanized honeybees in Arizona. We sponsored research to evaluate new technology, and an economist that is affiliated with the University of Hawaii is currently doing an economic analysis. And I will just go through some pictures. These are pictures of the early stages of the program when we were training the monitoring crews, the different invasive species committees, and even the vector control workers on Maui for identifying mosquitoes. These are training airport staff because they are our eyes and ears when the crews are not there to monitor the traps. So we will train landscapers and even companies that that go do trimming of the palms at airports. Our traps are mainly located in secure areas, even though some of them are in just regular areas, but most of them are in secure areas because these areas are where we could potentially intercept a new species, like for example, cargo areas or luggage sorting areas as well. As I said before, the program sponsors training in Arizona so that we can learn more about Africanized honeybees, so we can get back and work on an action plan for Africanized bees. And we are also working on some other deliverables for the Department of Transportation, putting together best management practices, BMPs for contractors and workers, so that they make sure they don't bring any potential invasive species to the airport. Like, for example, they finish a job in Waimanalo where there might be little fire ant in there, and they take that same machinery to the airport, and they can potentially bring LFA with them. So we are kind of laying that out in these BMPs. We are developing language for contracts so that DOT can enforce those BMPs. And finally, the economic analysis, I think, is going to be a really great piece of information. So it compares the cost of monitoring versus the cost of inaction. And it's already showing that there is certainly economic benefit of doing what we are doing. And it will save money to the state because yeah, these invasive species can be really have very important impacts in the economy. And this slide just shows a little bit of the preliminary results of the economic analysis from Donna Lee, that is the economist that is helping us with that. And for example, the cost for the surveillance of CRB at all 6 airports is estimated to be at $30,000 a year. So if we don't do this monitoring and we just let CRB kind of like do its thing, if we are to respond while they are already doing the damage and we have to mitigate that, the cost will be 6-fold. And if this it gets really out of hand, and then we start seeing really extensive damage of palms and that need tree replacement, then we are looking at the cost being 18-fold. So, and just to wrap it up in time, so the program benefits are many. So basically, we are able like that to detect and these target invasive species, avoid costs to the state. And I think one of the main things is also we are trying to develop this strong collaboration between different agencies to be able to have more biosecure Hawaii. Future of the program, we are in a last year of funding from DOT, so we are looking for alternative options for continuing with this this program. Actually, I will be jumping off this soon, this talk, and then I will be joining a meeting regarding funding for this program. So, hopefully everything goes well and we can continue with this program. We would like to add new targets probably in the future as well, and especially expand this program to seaports, and maybe even establish some collaboration with other states or islands in the Pacific region. So we are looking for the same things they are looking. And this slide just shows the air traffic worldwide. And then if you look at here in the middle, in the Pacific Island, you see a lot of air traffic here to the Pacific, to Hawaii. And this is actually projected to increase by 2036. And this is the same thing, just showing the maritime vessel movement, and you can see that in the Pacific we are really a hot spot there, and all these kind of routes, all these arrivals are a potential arrival of a new invasive species as well. So that's why we want to also expand this program to seaports. And with that, this is my last slide. It just shows the planning team and the implementation team. So really big thanks to all these organizations and people that basically had all those meetings prior the launch of this program, and all the people that are already part of the implementation and part of the program. So with that, I am done with my presentation. And I will stop sharing my screen right now, if I may.
Yeah, mahalo, Leyla. That was amazing. What a massive effort that is, and just multi-agency statewide biosecurity. So just great to see that program and what you've intercepted and what everybody's been able to do together with that. At this time, we have A few minutes for questions, so please feel free to ask your questions either in that Q&A or you can go ahead and put them into the chat box. I already see a question here for you, Layla. Any concerns about the genetically modified mosquitoes? I'm assuming like the Aedes aegypti programs that are happening on the mainland.
I think there is some efforts here for that as well. For the, I guess maybe the person is referring to the Wolbachia stuff. I'm not sure, but this is not part of the program. But certainly, this is a way to get rid of mosquitoes. So I am actually excited to see that there's programs going on to try to manage and get rid of mosquito populations in Hawaii, because they are not only a public health risk, but also they can have impacts in our native fauna here in Hawaii. So yeah, they're not part of the program, but this is something that the DLNR is looking into.
Yeah, thanks, Layla. And I think Leah put that question in. So if that wasn't where you're going, just feel free to clarify that in the chat box.
And there is some groups doing some work, so we can definitely, if you reach out, we can connect you with those groups, and you can get your information from there as well.
Yeah, and maybe just a point of clarification on the Wolbachia, that's actually a bacteria that already exists in insects. So it's not a genetically modified organism at all. And that's kind of the route here in Hawaii we're taking with how to reduce mosquito numbers and potentially save native forest birds by using this Wolbachia bacteria. Yeah. Layla, I have a question for you while we wait for other people to kind of chime in. You know, if you expand to seaports, which of course is such a major pathway for other pests to come in, like what would be the focus or what would be the priority pest or how would you come about figuring that out?
Well, I think there is already some sort of like surveillance happening at seaports, but I think the main gap in there will be the mosquito pollution, because, for example, our partners from the Department of Health they have been asking for all these years, like if we can kind of like support them at seaports, but due to the nature of the funding that we have right now, we can only focus at airports, because the money is tied to airports. So that's why we are really kind of like looking at other ways that we can fund this program. So we are not tied to the targets that we have right now. We can expand, but also we can expand it to seaports. So, but yeah, so if there is a priority for which species we will prioritize, probably will be mosquitoes. So setting up more traps for mosquitoes at seaports, because that's how we have been doing it even for airports, because we don't really pay for the manpower for the vector control. They already restore the vector control branch. So what we are doing in terms of mosquitoes is basically adding the capacity in terms of traps, because they had some budget issues and the traps that were using, the numbers were not appropriate, or even the type of traps were not the ideal. So yeah.
Yeah, thank you. Let's see.
I think there's another question in
Yep,
there. How
we
does
got,
Māmalu
oh, go ahead.
So how does Māmalu Poepoe differentiate between HDOA inspection centers? Is there collaboration there? So yeah, so just to kind of like tease that apart. So Māmalu Poepoe is a surveillance program, whereas the HDOA is, and the inspection is like when you go through what comes here and then you inspect. So like what Māmalu Poepoe does is the surveillance at ports outside. So hopefully whatever misses that inspection can get into our traps. And we coordinate our efforts with the Department of Agriculture. For example, they did have, for example, swarm traps and CRB traps at airports already, but they had it mainly at the unsecure areas at the airport, but we added a bunch of them in the secure areas where there is more difficult access. You have to get a badge. And that's when we came in coordination with them, because they suggested, okay, I think this area of the airport is that we really need it. We need more surveillance because our people cannot get there. So that is the collaboration part. So, and the other question is, how much surveillance is done with shipping agricultural items to, say, large stores that have been known to bring in ants in potted plants? I think this is more kind of the inspection side of that, question that in my point of view is linked to the previous one. So this is kind of like when you are shipping things, hopefully you can catch them through inspection, but again, like, so we don't see that part of the inspection part, because this is done by plant quarantine, the inspectors there. But if anything kind of like gets out there, so that's why we do the surveillance with the baited vials, so that we can detect those in there. And I don't see any more questions. Do you?
There is another question in the chat for you.
Yeah, is there?
What are the target species that would be potentially surveyed for in the future?
Well, yes. So in the Department of Agriculture, for example, suggested the Japanese beetle in there. So we definitely will be considering targets based on what our partner agencies feel that are an issue. So, but that species, I think, is certainly in the radar, and we currently don't conduct surveillance for that. So we are hoping to maybe in the future start talking about that, and maybe train our partners to monitor for those species.
Yeah, I think that's all the questions. And I think you're warmed up for your funding discussion now.
Yeah. All right. I will have to jump off, but I will try to jump back if my funding discussion goes well or on time.
Sounds good. You're among friends here. Thank you. Mahalo, okay. Leyla.
Bye.
All right. Thank you, everybody. Those are great questions. I always love the discussion. We'll get into the second part of our biosecurity series. We have Mark Wright with us. He is a professor of entomology at the University of Hawaii at Manoa in the plant and I always get acronyms all messed up. Plant
and Environmental Protection Science.
Yes. Okay. I was thinking of what the E was. Thank you, Mark. He has an extensive background in biological control research and is a really interesting project using bee pheromones to manage elephants. He's born and raised in South Africa, and we're really lucky to have him here to talk to us about the successes of biocontrol here in Hawaii. So again, you can type questions into the Q&A and into the chat box along the way. And with that, I will hand it over to Mark to share your screen.
All right. Thank you, Chelsea. Yeah, well, thanks for the invitation to be here. It's great to join you all and to be able to talk about biocontrol in Hawaii, and particularly the successes that we've had. So I'm going to talk not so much about individual case studies and their successes. I will talk about some that are really kind of interesting, I think. To all of us, but I'll also talk quite a bit about the science that goes into biocontrol and making it safe in Hawaii and making it successful. So let's have a quick look at biocontrol of insects in Hawaii in a historical sort of perspective. We have one of the longest histories of biocontrol in the world. Initially started out by various private people and organizations, and then taken over by the Hawaii Department of Agriculture. Who until today they conduct the vast majority of biocontrol projects in Hawaii. The first introduction was back in 1890 when a predator of cottony cushion scale was introduced from Australia to Hawaii, and was highly successful. It was highly successful because it attacked the target pest very efficiently. It didn't attack other insects to any extent, and that spawned a lot of interest in using biocontrol agents. And that's a predator, is Rhodalia. It's a ladybug. So plenty of ladybugs or lady beetles were introduced into Hawaii around that time, the early 1900s, and it was done fairly indiscriminately. A lot of that was not at all effective, and some of them attacked non-target species, native species, of course. And a lot of these early introductions were done by people who were not really biologists. They were well-meaning farmers and farmer groups and so on. We're still badgered by historic issues like that, like the mongoose. You're all thinking about that, I know, and so on. That doesn't happen anymore. There are plenty of success stories. We actually have a very high success rate in Hawaii compared with the rest of the world. And ultimately, many invasive insects are very effectively suppressed by biocontrol, and many weed species are suppressed. I don't really work with weeds extensively, so I'm going to speak about insects for the rest of the talk. I have some colleagues at the Hawaii Department of Ag who do some great work on weeds, Forest Service over on the Big Island. So there's a fair amount of weed biocontrol going on as well. There was that period of negativity where people were really unimpressed with biocontrol in Hawaii. And this was particularly from the mid-1980s until more recently, when there were some researchers at, for example, the Bishop Museum in Honolulu, who pointed out or who suggested that a number of native species of insects had been driven to extinction by biological control agents. And this caused a great stir. There were papers written in very influential journals like the Annual Review of Entomology and so on, which received a lot of attention. And this caused a lot of negativity towards what we call classical biocontrol, which is when you go to the place where a pest comes from, find a natural enemy and bring it back here, put it through a quarantine period, make sure it's safe, and release it today. Perhaps in the early 1900s, not so much. Those are the ones that caused the concern. And that impacted the way biocontrol is done today, by the way. It's very strict today. And I'll get back to that later. Back in the early 1900s, and it sounds strange to say that now, doesn't it? Back 1950s, 1960s, if you got a potential biocontrol agent that you wanted to bring here and you found that it did attack a natural enemy, you think, well, that's great, because a native species. The native species can provide a kind of a reservoir to sustain the natural enemies when pests were rare. That would never happen today. That's a practice of the past that's gone. There were issues back then, right? In the early days of biocontrol, there was no real risk assessment done in many cases until the 1970s, really. There was very little post-release monitoring. It's an expensive thing to do, and it's very hard to actually sustain that. And in Hawaii, there was a lot of concern for endemic species being attacked by biocontrol agents, which was taken to heart by, I think, everyone working in biological control. The Hawaii Department of Ag has extremely strict procedures in place that ensure that biocontrol agents are safe to introduce, as do Fish and Wildlife, Forest Service, and all of us involved in biocontrol are very careful today. When non-target screening is done at the Hawaii Department of Agriculture, what happens is that insects that are considered to be potentially useful biocontrol agents are brought into the quarantine on Oahu. And there they're kept for as long as it takes to conduct the screening, which can be years. A number of potential non-target hosts, either native species or beneficial insects that have been introduced, are brought into the quarantine, and then tests are done to see if that potential biocontrol agent will attack them. And it's a very conservative process today. Essentially, if there's any non-target attacked in the quarantine, that potential agent will be rejected as a possibility, which has its issues, but it also results in extremely safe biocontrol. And in fact, following that very strict approach, since releases made in 1975, there's not been a single non-target attack recorded out in the field, which I think must give one some faith in the procedure now. That's a good, it's longer than 40 years, right? With no attacks. There are recorded attacks, of course, which occurred from some species that were introduced subsequent to 1890 some and up to 1972, let's say. What this graph shows is the years on the horizontal axis and the number of non-target species attacked on the vertical, the y-axis, and the number attacked of those that have been detected over the course of multiple years. So the species that was introduced in the late 1890s, it was a parasitic wasp introduced for control of caterpillars in sugar, I believe. It attacks 20 known non-targets. And so there are a number of others that attack 1 to maybe 4 non-target species. And then there's one with a question mark here, is Meteorus lefigmi, which was actually introduced back in 1942, but maybe, or maybe did not establish, and was reintroduced in 1972. And that is the last known introduction to have non-target impact. So we've had 45 years now, I guess, of safe biocontrol, and plenty of other safe introductions. Look at that long line down there of zeros. Okay, that's important to notice. I think there's only 12 species that were recorded to have non-target attacks. Of about 283, 284 species that were introduced over the years, 115 of those have become established in the state, and they continue to suppress pests. And a few of those, as you can see in the graph, have attacked non-target species. The extent of those impacts is debatable. So just quickly, let's look at the level of success that we've had with biocontrol in Hawaii. Globally, the level of success in classical biocontrol is averaged around 18%. In Hawaii, our success rate is about 35%, which is quite remarkable. It's pretty much double the rest of the world's success rate, probably because of the huge effort that's been made in the state and the careful work in selecting potential biocontrol agents, I think. There are a couple of ways to measure that success. What can happen is the pest may be completely suppressed, that it's no longer really a pest. It doesn't cause any economic or ecological damage. And that's wonderful. And a good number of the introductions we have in fact resulted in things no longer being pests at all. But if it doesn't happen, if you don't completely remove the pest as a problem, you might measure success as the fact that you reduce dependence on pesticides, for example, or other means of managing pests. Either way, the economic impacts of those invasive species and the ecological impacts of them being around, for example, using insecticides, are reduced and can be extremely beneficial environmentally, economically, and in a health sense, human health sense. So let's look at some of these cases of non-target impact that have been analyzed over the years. A good few have. Leyla, who just spoke, she did a PhD on non-target impacts, and I'll talk about some of her work later. Here's a piece of work that was done by Tracy Johnson and others, looking at an endemic bug, the koa bug, a spectacular animal that I'll show you in a moment, an endemic bug that occurs on koa and a few other things in Hawaii. Back in the 1960s, Nesera viridula, this little bug, these guys that are sitting around what looks like a little peanut bar down here, right? They're feeding on a peanut that we gave them in captivity. A couple of parasitoids were introduced to control them. Trisulcus, which is a parasitic wasp that you can see in the top right bubble there. You can see a bunch of them popping out of the eggs of the green stink bug. And then also Trichopoda, this fly. 2 species were introduced to Hawaii, and they parasitize stink bug adults. Trisulcus parasitizes the eggs. All right. Now, oh, here's a picture of the koa bug. Stunning creature. Great images here from Nate Ewan. It's a beautiful little animal that you can find if you go and look around on koa. You can find them in haole koa as well, for example. It lays these stunning eggs in little patches that are pretty similar to green stink bug egg masses. And they're used in captivity quite readily by parasitoids. In fact, back in 1964, when the parasitoids were going to be introduced, people thought, oh, okay, koa bug could be a useful non-pest host and keep the biocontrol agents going in the field. Now, as I said, that would never happen again. That was back then. This is now. So the koa bug occurs on its native hosts, Acacia koa, of course, it's its main host. It also occurs on Dodonea viscosa, which is a pretty high elevation shrub that you can find on some of the islands. It also uses invasive species, Acacia confusa, down here. You can see it in the image down there. And it'll also occur on Lucina, Hawaiʻi koa. There's quite a wide distribution from the lowlands right the way up into subalpine areas. It occurs in dry forests, it occurs in mesic forests. And this produces a lot of gradients in habitat and environmental conditions over which they occur, and which can obviously influence the parasites that we're looking at. So Tracy Johnson and others published this work back in 2005 in Ecologia. They looked at the impacts of those parasitoids on koa bugs by very carefully analyzing what we call life tables, which allow you to calculate the probability of an individual dying from a particular cause. And this is very intensive work. It requires a lot of very careful monitoring of insects and a lot of counting. And it produces the most accurate results possible for analyzing impact of a parasite on a generation, on the generation of multiple generations of insects. What they showed is that Trichopoda, the fly, had pretty much zero impact on koa bugs. And they found that that impact was very highly density dependent. What this means is that at low densities, there's very low impact. At high densities of koa bug, there'll be higher impact. Okay, there's a correlation between density of the host and the impact that the parasite has. So under typical koa bug densities, the impact is very low. It's there, but it's relatively, it's low. The effect of the egg parasitoid, that little wasp that attacks the eggs, Trisulcus, is low. But that just very distinctly depended on elevation, and it depended on habitat that they were occurring in. You could get up to 26% parasitism, depending on where they were. So what the life table analyses showed that at low elevations, Trisulcus, the egg parasitoid, was producing the highest level of attack on koa bugs. There were also invasive predators, ants, and a few other things that were causing a lot of that damage. At higher elevations, it was really just predators, egg predators, and they're all invasive. None of them were introduced as biocontrol agents that were causing the damage. So the impact from Trisulcus at higher elevations is minimal. At low elevation, there may be some impact under certain circumstances. The interesting thing is that the highest parasitism that was observed was actually on the invasive plant, right, on the invasive host that koa uses, Acacia confusa, and on native plants it was essentially zero. So perhaps in places where Acacia confusa has replaced native hosts, you can expect some impact from the parasitoids, but in natural habitat, it's very unlikely to have extensive impact on the populations. And again, just to emphasize, egg predation from things other than the biocontrol agents, introduced ants, accidentally introduced ants, occurred across a whole range of elevations and a bunch of hosts, and is more problematic than the biocontrol agents turned out to be. So that's an encouraging study. And some other work that we did, and this was part of Leyla Kaufman's PhD work with me, is whether we can actually predict non-target host range within Hawaii from looking at the host range of the insects in the native range. So if we go to the place where we're going to bring a biocontrol agent from, can we make some predictions or forecasts as to what they might attack once they get to Hawaii? So we looked at this little fairly nondescript little moth. This is the caterpillar here. It's called Udea stellata. It occurs on mamaki, also the host plant of Kamehameha butterfly. And we discovered over the years that this little caterpillar, it's an endemic native moth to Hawaii. It's attacked by a whole bunch of parasitoids, 7 species in total. Some of them were introduced as biocontrol agents, and some of them were accidental introductions, and one of them maybe a native Hawaiian insect. So our interest here was to see if we look at one of those species that was purposefully introduced here or accidentally introduced, because we might be able to obtain data, could we have predicted, looking at parasitism in its place of origin, what it would do here? So what we did is, and I'm going to explain this very briefly, I don't want to get into too much detail here, but we looked at how likely it was that this particular wasp, Coleotichus marginiventris, would attack the native species here, Udea, based on what we saw in its place of origin. And we could go back to the literature and look at what it attacks in its place of origin, right? So it attacks there at least 6 species that are known. 4 of them are noctuids, that's in a different family. 2 of them are in the family Crambidae, which is the same family as Udea. So that could immediately raise a flag, like, oh wait, this thing might be a problem where you intend to introduce it. It was brought here to attack noctuid moths, which are armyworms that were attacking sugar back then. And if we look at the probability, the vertical axis on this graph shows how likely it is for a certain level of parasitism to be seen. And that level of parasitism rate is shown on the y-axis, right? So for example, 5%, 0.05 of the insects parasitized, expressed as a percent, would be 5% parasitism. The probability of that occurring in the place of origin is very close to 0. It's just a little over 0, and very similar in Hawaii. What you see essentially is that the pattern is very similar overall, right? There's a fairly high probability of fairly low parasitism in Hawaii, relatively close to what it was in the place of origin. And then for any higher parasitism levels, it was very comparable to the place of origin. Very close to 0 in the case of the species. So the potential that you'd have 15% parasitism is just slightly more than 0. And in fact, you could predict this from looking at the situation in the place of origin. I hope that makes sense. Please feel free to ask if that's confusing. So at least for that species, it seems reasonable that you could make those sort of predictions. And we did look more broadly, and obviously it depends how much data you have, but overall it looks like potentially, yes, if you have the time, money, and people to do it, you can potentially predict from the place of origin what might happen here. It's not 100% prediction. Another interesting thing, and it's a concern that a lot of us have, is whether when you introduce a biocontrol agent, does it evolve in this place, right? Evolution happens no matter what. And so if you introduce them here, do they evolve? Do they undergo what you might call an adaptive host shift to use new hosts? So if you introduce a parasite here from another country, will it evolve the ability to use a native species? The research that we conducted here was on a wasp, a parasitic wasp that was introduced from Australia into Hawaii to control fruit flies. And it started to attack a gall insect, a fly in the same family as fruit flies, but it causes galls on the stems of lantana, an invasive weed. Very different biologically, at least in the way, the place it lives. It's not in a rotting fruit like a fruit fly, it's in a hard case, the gall on a plant. And Diacasmomorpha, this parasitoid from Australia, started to attack the gall fly, which is originally from Mexico. So they had no evolutionary history. So our question was, did the wasp evolve the ability to use the gall fly, or was this just something that it was in its repertoire of capabilities? Perhaps in Australia it did utilize gall insects unbeknown to us. Some earlier work by some of my colleagues showed that yes, perhaps the wasp has developed a preference for the gall fly. Perhaps it's evolved that preference here. So we measured whether that change in behavior and host range could actually be seen in genetic change, in other words, evolution in the insects. I've kind of summarized some of this already, haven't I? The wasp is from Australia, no previous evolutionary contact with the non-targeted attacks here. It was introduced in the early 1900s, a very small introduction. I think only 14 individuals were introduced. By the 1930s, it was found in those galls, and probably forced to do so because of competition with another fruit fly parasite that was introduced. What we did was go to Australia and collect wasps, bring them back to Hawaii, and we could raise them in captivity. We had a permit to raise them. They were already introduced, so it wasn't so hard to get. And we could go to the Bishop Museum and get historic specimens from back in the 1900s across the years in nice chunks of time that correlated with things that we were interested in, like when did they move onto the galls. We could rear them from gall flies here, and we could also rear them from fruit flies. So we could look at their genetics across that whole period of about 100 years. We could extract DNA from them and sequence that, and then you can analyze the evolution of those organisms based on the DNA by reconstructing the relatedness, the phylogeny. And these are the results. It's kind of a cool shot on the right here of two Diacasmomorpha actually parasitizing those galls. You can see they're inserting their ovipositor, their sting, into the gall there. A very hard structure, very different from probing in a fruit. These are the results, and again, kind of briefly. And this is kind of like a family tree for those not familiar with phylogenetic trees. It shows the evolutionary history of species or organisms. And the closer they are together on here, the more closely related they are. What we're showing here is Diacasmomorpha trianae, in both cases, the parasitic wasp that was introduced that we're interested in, and the comparison that we made now. Okay, these are ones that were obtained in 2005, 2008 from Australia, New South Wales. You can see they are a little different from those from Hawaii. All of these in the middle here are from Hawaii. These are the ones we raised from the gall insect. These are ones that we had from 1948, 1961, 2004, 2007, I believe, our lab colonies. Historic samples from Australia, and they were collected from various islands. And there's no difference between those, interestingly, right? They have diverged a little from the ones that are in Australia now. There's been a bit of what we call genetic drift over time, but there's no genetic adaptation, no evolution in these wasps to attack the gall fly, which is encouraging for us. It suggests that even though this organism has undergone quite a dramatic change in the biology that we observe, it's not been an evolutionary change. And in looking at the evolution of biocontrol agents across the broad spectrum, some work I did a couple of years ago, again, we found that there was no adaptation of biocontrol agents to new hosts. There were those that were generalists, right? And they could utilize new hosts that didn't require evolution. And biocontrol agents have evolved to adapt to different climates and so on, but not to non-target species. And in general, that's really encouraging for us. I'll just briefly look now at how successful introductions are to Hawaii in terms of actually establishing and how well things are going. We have remarkable records in Hawaii from 1890 with the Hawaii Department of Ag. They have little cards that were written up way back then. It's amazing to look at these things. And they include data such as number of individuals released, places they were released, and whether or not they established. And that's a really interesting thing. Did they or did they not establish? It's something we're really interested in analyzing, is how do you make establishment more effective? So we could analyze these factors, and we're still doing this in extreme depth now. But I'll just talk really quickly about some preliminary data. So propagular pressure, the number that we introduce, or the number of times we introduce them, does that make a difference? Does it matter if they're host specific or not? Do they use alternative hosts, whether they're native or invasive? And do those things drive whether they establish or not? What we're finding is that number of other pest species is the biggest factor that actually drives establishment. And this is just using multiple regression analyses, which we don't have to go into here now. But this is what we call facilitation, right? So if there's a host that they can utilize here, it's another introduced species, happily, you're more likely to have establishment. It's not the same for native species or indigenous species that they utilize. In fact, that was slightly negatively correlated with establishment. So the more of those that they attacked, the less likely establishment was, apparently. And overall, if they're host specific, they're less likely to establish, which is a bit of a sad thing because we seek monophagous or host specific species for biocontrol. These graphs just show on the x-axis, in this case here on the white one, the number of species of parasitoids. These are just parasitoids introduced, and the odds of establishment. So how likely were they to establish, right? So, was it a 3-to-1 likelihood, or a 1-to-1, or a 1-to-0 likelihood? And all I'm showing here is when they attack other species, the more they attack, the more likely they are to establish. However, if they do attack native species, it doesn't actually improve the likelihood of their establishment. In fact, it decreases ever so slightly the more they attack. We're still analyzing this to build in a whole bunch of other variables to try and understand this better. The yellow graph on the right here is just looking at how number of individuals released is related to percent of species established. And what we find is that the more that are released, if you get to over maybe 500 to 1,000, you see a dramatic increase in the likelihood that they will establish in this environment. It also seems the more places you release them, the more likely they are to establish. And now, what I want to talk about quickly towards the end here, I hope I still have enough time to do this, is one of our real nice success stories of recent, which was a collaborative project between the Hawaii Department of Ag and us at the University of Hawaii and a couple of other institutions. So erythrinas are these trees that used to be planted as ornamentals and windbreaks in Hawaii. We have a native species, Erythrinus sandwichensis, that is an endemic to Hawaii and occurs in dry forests. Now, usually they have nice normal leaves. What happens if the invasive gall wasp, which came from Africa, attacks them is the wasp lays eggs into the leaves, and the larvae produce a chemical that causes this galling. It causes these massive lumps on the plants, which are kind of almost cancerous. And eventually the leaves will drop off, and eventually plants will die from this. They have dramatic impacts for tiny, tiny, tiny wasps that are attacking these plants that can cause monstrous effects. In fact, to the extent that the endemic species, the willy tree, Erythrinus sandwichensis, was considered to be threatened with extinction. People were banking seeds and so on because it was anticipated that it would become extinct. Mohsen Ramadan from Hawaii Department of Ag found a parasitoid in Tanzania. We found the same species in various other parts of Africa and imported those into quarantine, and within a couple of years completed the quarantine screening and had them introduced into the environment in about 2008. This is a nice, neat project that has allowed us to measure the impacts of biocontrol over a long period. We have 10 years worth of data, and I'll show you really briefly some of the outcomes. It was highly successful. We saw within a year that the numbers of gall wasps and their impacts were reducing. These are some data here just from 2013 to 2018. And we changed the way that we monitored things between 2008 and 2013, so the data are divided into two chunks. But across those months, which is, 5-6 years, this just shows the change in damage. The vertical axis shows the damage rating, which went from 0 to 3. A 3 would be like I showed you back in that first slide of the impact, and a 0 is, of course, no impact, no damage. And what you see is from around about 2013, where the average was probably around just under 2, the rating, it's dropped dramatically down to now where it's in the region of 0 to less than 0.5 on average, which is quite remarkable. This is the leaf damage. We do see that some of the areas that are drier, it's been slightly less effective. So this is on West Oahu, Makuakea. You see over here, you have a little bit of damage still. There's a little bit of damage over on the Big Island, for example, up in the Waikoloa dry forest. That still prevails to some extent, but the trees are doing quite well. The damage has dropped dramatically overall, which is super encouraging. This is a very successful case of classical biocontrol in terms of reducing the pest. One issue, though, is that the trees are still struggling to produce mature seeds. This graph shows from 2008, when the biocontrol agent was first released. It's a little parasitic wasp, by the way, that we introduced, right? How flowering, the black dots are flowering, how the percent of trees that are actually able to flower has increased, and the percent of them that were maturing seeds over that time. And you see it went from 0 trees that actually produced mature seeds back in 2008 and a few years prior to that, very quickly to just under 20%. And it's increased to, well, that was back in 2011, about 65 some percent. I think it's probably up around 80 some percent now, that, or even more, 100% that'll flower. And seed maturation is a little bit lower because you still get attacks on the flowers by the gall insect. That still cause pressure that reduces the seed maturation. But there are seeds being matured out there, which is very encouraging compared to a few years prior to that data point being collected. The biocontrol is reducing damage on the plant, and it's allowing them to produce seeds. There are other pressures on those seeds, sadly, but we don't have to talk about that right now. And today you can go out. This is in the Waikoloa forest. 2018, I think, or 2019, I took this image. You can go out there and find trees that are looking really good. They're bearing foliage. There's very low gall damage, and they're prevailing. And this is true in most places you go and look at willy trees. So aside from being really good for the willy trees, that project also showed people that classical biocontrol, which is often considered like an evil practice, can actually contribute effectively to conservation efforts, which is great. A lot of people who were previously who worked in conservation organizations and were displeased about biocontrol actually changed their attitude substantially, and they were very supportive of the practice. And overall, in a way, the impression around biocontrol was improved to a large extent, at least back then, and we're trying to keep that going. Okay, overall, if we do, this is the last slide. If we look at the benefits of biocontrol, what it produces for you is a sustainable option for managing or removing a pest problem. If classical biocontrol works, you only have to do it once, really. You can introduce the biocontrol agents into the field, and they'll give you sustained pest suppression. There's a super high benefit to investment return. It's huge. I mean, it's unrealistically huge in some cases, particularly in agricultural crops, of course. But for investing a very small amount of funds into doing biocontrol, you can get huge economic returns. It reduces pesticide dependence, which is a plus all around. And overall, what it does is reduces the environmental impacts of invasive species, whether they're insects or weeds. Weed biocontrol can have amazing environmental impacts. It's been shown worldwide to be incredibly valuable. Okay, that was the last slide. If there are questions, I'd be happy to take the questions.
Yeah, feel free to type your questions into the Q&A and the chat box for Mark. Mark, thank you for that. Oh man, such a process. And I think you're only we're able to really dive into one half of the process, because there's that other half of going to these other countries where these pests are native to and trying to find pests of the pest. And it's just a whole nother laborious process to go through. So it's a really incredible projects going on with biological control, really intensive and really great people here in Hawaii working on biological control, like Mark and others from Department of Ag and USDA Forest Service.
Yeah, no, they're great people. They're super dedicated, yeah. And the exploration work is not necessarily just tedious. It actually can be good fun. It's amazing stuff.
Yeah, I imagine you're going to some off-the-grid places.
Yep.
We do have a couple questions here. Let's see. The first one is, do you think the coqui frog will ever be eradicated? And maybe this is in the context of like, is there any biological control possible for coqui frogs?
Yeah, well, so just by the way, biocontrol seldom eradicates the target. Just to mention that. So I know some people are looking at fungi. There's a chytrid fungus, I think, that kills frogs, and I think some people are considering that as a potential coqui frog biocontrol agent. And there's some worms like helminths that get into the gut, and I think they can cause malformations, and I think some of those are being looked at. I don't know how successful they're being, and I don't know how easy it'll be to actually implement that. I don't think it'll ever be eradicated from Hawaii, no. I mean, perhaps if we had an invasion of tree snakes from Guam, perhaps they'd take them out. But yeah, I'm not very optimistic that that's a good biocontrol target.
Yeah, thanks, Mark. That's a good point, and I think it's just important to, you know, try to prevent it from spreading and becoming established on the other islands, like Mark was saying, Hawaii Island. You know, that's going to be really hard to even minimize the numbers there, even with a biological control, if it ever becomes useful.
Right.
But if you hear coqui and you know what it sounds like, and you're on one of the other islands, or even in some place outside the east side of Hawaii, call that 643-PEST or go to 643pest.org. That's what Elizabeth here manages, and we can help you out. Okay, another question from Monty. Do you have any successful biocontrol introductions of an invasive ant species elsewhere in the world that might give us hope regarding control of ants here in Hawaii?
Ants are really difficult to biocontrol. So people have tried using phorid flies, the little parasitic flies that attack the ants and lay eggs into them. They've tried introductions of those on the US continent, and I don't think it's ever been resoundingly successful. I think it's been somewhat promising in some areas. I don't know of any single case where ants have been effectively biocontrolled, though. No, they're resilient to that.
Yeah, they're tough buggers. Let's see, we have another question. Lori, have you noted that any biocontrols, specifically insects, have traveled from island to island on their own? That's a good question.
Yeah, there are some, and I'm actually struggling to remember which ones they were right now. So we have some trichogramma here, which are they're tiny egg parasites, and I believe some of those have actually been able to get from island to island on their own volition, because they're tiny and they get blown in wind currents, I think. And there are a couple of other examples. It does happen. I mean, particularly with things that get moved. So I think the one parasitoid of papaya mealybug, I think, started to spread between islands, perhaps because of human intervention, moving infested produce, but they seemed to move at some point without us moving them. We did eventually move them because they were useful. Yeah, it can happen pretty easily, I think. I think it happens more than we realize. There's also a question from JB, I see in the chat. JB asks, he says, I assume it's okay if there are non-target effects of biocontrol and other species attacked that are also invasive. He's thinking of Secusia, right, that was brought into control. Fireweed, but attacks German ivy. Yeah, yeah, absolutely. I think it's kind of a bonus. In fact, in that case, I think the HDOA knew that Secusia would attack the ivy. Yeah, I think it's been more effective on ivy than it has on fireweed, actually.
Wow.
Yeah, so you know, you kind of avoid this that what we call oligophagy. You avoid that in quarantine because it's maybe an indicator of potential non-target impacts on native species once it gets out. But if you can find things that will only attack a small range of pests, I think that'd be great. I mean, they're more likely to establish in the environment, according to the data we have.
Mark, I have a question. What is the timeline from, you know, the start of trying to find a biological control agent to like release? Like, what's the typical timeline?
It can vary quite dramatically. So the Erythrina gall wasp project, the Willy gall wasp project, was quite remarkable in that we were able to, and there were a bunch of us out there looking for natural enemies. I think within a year we'd found the natural enemy, got it into quarantine, and I think within 3 years it was out in the environment, which is remarkable. That's kind of one of the fastest ever, as far as I know, under the current conditions. But usually it takes longer than 5 years. It'll take maybe 5 to 10 years in some cases. I know of some species that have been in quarantine for maybe 12 years.
Wow.
So it depends, if there are concerns with the non-target screening when it comes to Board of Ag review and so on, they'll send it back for further work and so on. And that can be quite trying. But if we have more people and more space to do the work, we can move it along quickly.
Yeah.
I have a question related to that too, and hopefully it's not too complicated. But I was wondering, I mean, this sounds like a challenging process to do this correctly. But what is the biggest challenge that you see happening in the state right now with the implementation of biocontrol?
Oh, the biggest challenge, I think, without a doubt, is the availability of quarantine space. Our ability to work on multiple species simultaneously. So the Hawaii Department of Ag quarantine is old and small. I mean, they do great work down there, but they just don't have the space or the facilities to work on many species at once. It's a very small facility, and it's really old. The one with Forest Service on the Big Island is also small. It's targeted mostly at invasive plants. But again, it's small, there's no way that we can have HDOA researchers and UH students and postdocs all working in the quarantine currently. There's just not space available. That's really the biggest bottleneck. I mean, there are a bunch of other things I can complain about, but that's our biggest issue. The whole legislative process is painful, but, one just has to deal with that. Yeah.
Yeah, thank you, Mark. Quarantine does seem like a challenge, and having enough space to work on your insects and beyond insects, things that are smaller, which there's not the capability with the current facilities. But it is amazing the amount, like the success rate that has come out of the facilities with the folks that are working on it, the small number of folks that are dedicated to biological control work in those facilities. We also have Leyla back on, so she looks happy, so it must have gone well, and happy. We have a few minutes left. If anybody has questions for her or Mark, please feel free to put those in the chat or into the Q&A box. I know sometimes I can't type fast enough to ask my questions. And Leyla, we just realized that you did your postdoc work with Mark on biological control. So what a great co-presentation we had today.
And he was my PhD advisor as well, so.
That's awesome. You still like each other, that's good.
We put up with each other, yeah.
Yes.
Great work, really.
Yeah, I don't see...
Because of the guidance.
It's important. Yeah, I don't see much else in the chat. So, I mean, we only got 4 minutes left, but we can close it off early. And just want to say thank you to Mark and Leyla for presenting about these really great projects, and just Mark for helping us understand biological control and some of the misconceptions we tend to have about that, and Leyla for opening our eyes to airport biosecurity. Really great projects. And Elizabeth, thanks as always for helping out with HISAM webinar.